going to discuss about active and passive transport. Let us see what is passive transport. In order to understand how substances move passively across a cell membrane, it is necessary to understand concentration gradients and diffusion. What is a concentration gradient? A concentration gradient is the difference in concentration of a substance across a space. Molecules or ions will spread from where they are more concentrated to where they are less concentrated until they are equally distributed in that space. When molecules move in this way, they are said to move down their concentration gradients. What is diffusion? Diffusion is the movement of particles from an area of higher concentration to an area of lower concentration. A couple of common examples will help to illustrate this concept. Imagine being inside a closed bathroom. If a bottle of perfume were spread, the scent molecules would naturally diffuse from the spot where they left the bottle to all corners of the bathroom, and this diffusion would go on until no more concentration gradient remains. Another example is a spoonful of sugar placed in a cup of tea. Eventually, the sugar will diffuse throughout the tea until no concentration gradient remains. In both cases, if the room is warmer or the tea is hotter, diffusion occurs even faster as the molecules are bumping into each other and spreading out faster than at cooler temperature. Having an internal body temperature around 98.6 degrees Fahrenheit thus also aids in diffusion of particles within the body. Let us consider substances that can easily diffuse through the lipid bilayer of the cell membrane, such as the gases like oxygen and carbon dioxide. Oxygen generally diffuses into cells because it is more concentrated outside of them, and carbon dioxide typically diffuses out of the cells because it is more concentrated inside of them. Neither of these examples requires any energy on the part of the cell, and therefore they use passive transport to move across the membrane. As a result, oxygen will diffuse from the interstitial fluid directly through the lipid bilayer of the membrane and into the cytoplasm within the cell. On the other hand, because cells produce carbon dioxide as a byproduct of metabolism, carbon dioxide concentrations rise within the cytoplasm. Therefore, carbon dioxide will move from the cell through the lipid bilayer into the interstitial fluid where its concentration is lower. This mechanism of molecules spreading from where they are more concentrated to where they are less concentrated is a form of passive transport called simple diffusion. Here is a diagram to show how simple diffusion across the cell membrane occurs. Another type of diffusion is facilitated diffusion. Solutes dissolved in water on either side of the cell membrane will tend to diffuse down their concentration gradients, but because most substances cannot pass freely through the lipid bilayer of the cell membrane, their movement is restricted to protein channels and specialized transport mechanisms in the membrane. Facilitated diffusion is the diffusion process used for those substances that cannot cross the lipid bilayer due to their size or polarity. A common example of facilitated diffusion is the movement of glucose into the cell, where it is used to make ATP. Although glucose can be more concentrated outside of a cell, it cannot cross the lipid bilayer via simple diffusion because it is both large and polar. To resolve this, a specialized carrier protein called a glucose transporter will transfer glucose molecules into the cell to facilitate its inverse diffusion. Let us see what is the role of osmosis in this type of diffusion. Osmosis is the diffusion of water through a semi-permeable membrane down its concentration gradient. If a membrane is permeable to water, though not to a solute, water will equalize its own concentration by diffusing to the side of lower concentration and thus the side of higher solute concentration. In the beaker on the left, the solution on the right side of the membrane is hypertonic. The movement of water molecules is not itself regulated by cells, so it is important that cells are exposed to an environment in which the concentration of solutes outside of the cells is equal to the concentration of solutes inside the cell. Two solutions that have the same concentration of solutes are said to be isotonic, that is equal tension. When cells and their extracellular environments are isotonic, the concentration of water molecules is same outside and inside of the cells. Osmosis occurs when there is an imbalance of solutes outside of a cell versus inside of a cell. A solution that has a higher concentration of solutes than another solution is called to be hypertonic solution, and water molecules tend to diffuse into a hypertonic solution. Cells in a hypertonic solution will shrivel as water leaves the cell via osmosis. In contrast, a solution that has a lower concentration of solutes than another solution is said to be hypertonic, and water molecules tend to diffuse out of a hypertonic solution. Cells in hypertonic solution will take on too much water and swell, with the risk of eventually bursting. Another mechanism besides diffusion to passively transport materials between compartments is filtration. Unlike diffusion of a substance from where it is more concentrated to less concentrated, filtration uses a hydrostatic pressure gradient that pushes the fluid and the solutes within it from a higher pressure area to a lower pressure area. Filtration is an extremely important process in the body. For example, the circulatory system uses filtration to move plasma and substances across the endothelial lining of capillaries and into surrounding tissues, supplying cells with the nutrients. Filtration pressure in the kidneys provides the mechanism to remove waste from the bloodstream. Now let us see what is active transport. For all the transport methods described above, the cell expends no energy. Membrane proteins that aid in the passive transport of substances do so without the use of ATP. During active transport, ATP is required to move a substance across a membrane, often with the help of protein carriers and usually against its concentration gradient. One of the most common types of active transport involves proteins that serve as pumps. Let us see the sodium-potassium pump. The sodium-potassium pump, which is also called NK plus ATPase, transports sodium out of a cell while moving potassium into the cell. The sodium-potassium pump is an important ion pump found in the membranes of many types of cells. These pumps are particularly abundant in nerve cells, which are constantly pumping out sodium ions and pulling in potassium ions to maintain an electrical gradient across their cell membranes. Powered by ATP, the pump moves sodium and potassium ions in opposite directions, each against its concentration gradient. In a single cycle of pump, 3 sodium ions are excluded from and 2 potassium ions are imported into the cell. What is an electrical gradient? Electrical gradient is a difference in electrical charge across a space. In the case of nerve cells, for example, the electrical gradient exists between the inside and outside of the cell, with the inside being negatively charged around -70 mV related to the outside. The negative electrical gradient is maintained because each sodium-potassium pump moves 3 sodium ions out of the cell and 2 potassium ions into the cell for each ATP molecules that is used. This process is so important for nerve cells that it accounts for the majority of their ATP usage. Other forms of active transport do not involve membrane carriers. Endocytosis is the process of a cell ingesting material by enveloping it in a portion of its cell membrane and then pinching off that portion of cell membrane. Once pinched off, the portion of membrane and its contents become an independent intracellular vesicle. 3 forms of endocytosis are seen. Endocytosis is a form of active transport. First form, in phagocytosis, which is relatively non-selective, the cell takes in a large particle. Second form, in pinocytosis, the cell takes in small particles in fluid. Third form, in contrast, receptor-mediated endocytosis is quite effective. When external receptors bind a specific ligand, the cell responds by endocytosing the ligand. In contrast with endocytosis, endocytosis is the process of a cell exporting material using vesicular transport. Exocytosis is much like endocytosis in reverse. Material destined for export is packed into a vesicle inside the cell. The membrane of the vesicle fuses with the cell membrane and the contents are released into the extracellular space. Many cells manufacture substances that must be secreted like a factory manufacturing a product for export. These substances are typically packed into membrane-bound vesicles within the cell. When the vesicle membrane fuses with the cell membrane, the vesicle releases its contents into the interstitial fluid. The vesicle membrane then becomes part of the cell membrane. Cells of the stomach and pancreas produce and secrete digestive enzymes through exocytosis. Endocrine cells produce and secrete hormones that are sent throughout the body, and certain immune cells produce and secrete large amounts of histamine, a chemical important for immune responses. Thank you for watching our video.
तो कहाँ चले? लाइक और सब्सक्राइब तो कर लो। अभी कर ही दिया है तो बेल आइकॉन पर भी क्लिक कर दो। थैंक यू।